Good morning! So today we're going to talk about the prokaryotic replication fork. So right here I've drawn a bacterial circular chromosome, and just to show, we're going to be looking at this little excerpt of DNA right here. So here is our double-stranded DNA coming from that bigger circular chromosome, and we have to start somewhere, and that place where we will start is called the origin of replication, right here. And what will happen from that origin is, using DNA helicase, we're going to split those two strands and open up into a replication bubble with two forks, one right here and one right here. And I'm going to focus specifically just on one fork that I've depicted in this larger model. So as I said, the first step is we're going to have DNA helicase just breaking those hydrogen bonds to separate the two strands of DNA, while the phosphodiester bonds that are keeping the actual backbone, the sugar phosphate backbone, together are untouched. And then in order to relieve some pressure from unwinding those strands, we have a DNA gyrase just to make sure that the unwinding of the two strands doesn't happen too fast. And then, speaking about stability in the single strands, here we have little single strand binding proteins that make sure that we won't have any coiling or hairpins on that single strand of DNA. All right, so to begin our replication, I have shown here these little red squares are primers made by these little red primase enzymes, and so that's just a place for the larger polymerization enzymes to bind. So here we have DNA polymerase 3, which is going to bind to the primer, and since this is the leading strand, we're just going to replicate continuously from the 3' to the 5' direction, just straight through. So just adding those complementary nucleotides to replicate the strand. And now in our lagging strand, we're also going to take another DNA polymerase 3 enzyme, again binding it to the primers, and then we can just replicate a small strand and then another small strand called Okazaki fragments, because in the lagging strand we replicate discontinuously, again in the 3' to 5' direction, but discontinuously, so in these small Okazaki fragments. So we're not quite done yet. The primers aren't really part of the DNA, so we need to have our DNA polymerase 1 enzyme come in, and that's just going to take away these primers and replace them with DNA. So anywhere there's a primer, that's going to come in. And then finally, our last step is just to take these fragments, these Okazaki fragments, and with the ligase enzyme, just connect them, so that you make sure that you have a full and complete strand of DNA instead of a fragmented one. So there you have it. That is the prokaryotic replication fork shown by this model.